My dear friends, in my previous class, I have told you about the nature and sources of drugs. I have told you about mineral source of the drugs which are being used in therapy, that is, drugs obtained from the earth crust. Then I have told animal source, that is, some drugs are being obtained from the animals. Today also I will tell you some detail of few example of animal source drugs. Then I have told you about the vegetable source. Vegetable source mainly is plant source. That is, we are getting certain things which are being consumed by us as a food, and there are certain components which are obtained from the plants as a medicinal agent. These are categorized depending on their chemical structure. And then that's why they are classified under the different headings. Alkaloids, glycosides, oils, these are of three categories: fixed oil, volatile oil, mineral oils, resins, gums, tannins. These are different components obtained from the plant source and used in treatment of different diseases. Then biggest source nowadays, it is synthetic source, that is, drugs are being obtained by chemical synthesis. And fifth source is by growth of microorganisms. We are getting antibiotics, enzyme like streptokinase, modified E. coli growth gives insulin. In previous class I have told you alkaloids, glycosides and different oils. Now other components which we are getting from the plant source is resin. Now what is the resin? Resin is a component obtained from the plant as secretory product on the plants. They get accumulated on the stems and these are being collected and used for different purposes. Under this heading we can classify commercial resin, that is turpentine resin. It is being used commercially for making the paints. It is obtained from the turpentine trees. Then another resin which is used in therapy, it is resin benzoin. Benzoin resin, it is being used for preparation of a typical type of tincture. When a person goes in a hospital in emergency, having some accident and oozing blood from its body, then a brown color tincture is being applied that contains benzoin. Benzoin is one resin which is helpful in blocking the broken blood vessels. It forms a film over the broken blood vessels and the oozing of blood is being stopped. So this is another resin which is being used. Then benzoin resin or tincture benzoin is also used in the treatment of burn cases. In burn cases, this tincture benzoin is used, that is resin. Then another resin which is being used in therapy is balsam of Tolu. This is another resin which is being obtained from the plant, that is it is secreted by the plant stem and collected. Balsam of Tolu, this particular resin, it is a component of cough syrup. In other terms, we can say balsam of Tolu is used for suppressing the cough. It is for example, balsam of Tolu is a component of glycodine which is very commonly used for suppressing the cough. This is also one resin. So these are the different resin, we can say, which are being obtained from the plants. Benzoin, balsam of Tolu, then turpentine resin, its use is commercial use. It is used for preparation of the paints. Then next component is which we get from the plant is gums. Sorry, one thing more I want to tell about the resins. Resins which are being prepared in the plant automatically, they are polymerized volatile oils. Polymerization of volatile oils occurs in the plant itself, and then it is converted into resins, which is being used in therapy. Then gums. Gums are these are polysaccharides present in certain plants. These are also secretory product of the plant. You see, on the stem, gums are being, it comes exudate and is deposit on the stem which is being collected. Very common gum which is being used commercially also and in therapy also, that is gum acacia. Another is pearl tragacanth. These are polysaccharides, that is these are formed in the plant by, these are carbohydrates which are present in the plant and they are gums. These gums, that is gum acacia, pearl tragacanth, these are being used for preparation of different pharmaceutical components, that is preparation of emulsions, preparation of different suspensions. These components helps in preparation of these components which are used in therapy. So we can, in other terms, we can say gums are being used for as a um, helping hand in preparation of different dosage forms such as emulsions and suspensions. These are being used for that purpose. Gum acacia is nothing but where we commonly use it, gond. It is being used for that purpose. So this is another component which we are getting from vegetable source, that is the plants. The next is tannins. Tannins are non-nitrogenous component present in the plants. Non-nitrogenous component present in the plant which are used in therapy. Tannins are used as astringent. Detail we will go when we read the dosage form. Tannins acts as astringent, that is, tannins when applied or taken inside the GIT tract or applied on the burnt surface, their property is, tannins property is, it coagulates the cell membrane protein. Cell membrane protein is being coagulated by the tannins, and that coagulated protein forms a protective layer over the surface where it has been applied. In burn cases, it is applied over the skin. In GIT tract, it is being taken in the form of anti-diarrhea preparation, where astringent action of the tannins helps in reducing the frequency of diarrhea, frequency of defecation, because it forms protective layer inside the GIT tract, and the irritant component which is causing the irritation of GIT tract and which is creating problem as a diarrhea in the patient, that is being stopped by use of tannins. So these are the different components obtained from the plant source: alkaloid, glycoside, oils, resins, gums and tannins. I hope it is clear to you the drugs obtained from the plant source. The fourth category, which is very important nowadays, this is synthetic source. That is drugs are being obtained for the use of treatment of different diseases. These are prepared in the laboratory by chemical synthesis. Example is sulfonamides, aspirin, procaine, chloroquine, antimalarial drugs. These are being prepared in the chemical laboratory, and there are so many, long list is there, the drugs which are being prepared by the chemical synthesis, and it is a category we can narrate here. 60% of the drugs nowadays which are used in therapy practice, these are obtained from the synthetic source only. Other sources are still being used, but synthetic source is the biggest source now to prepare the different drugs to be used for the treatment of different diseases. Then comes by growth of microorganisms. This is also a big source of getting the drugs, and under this heading we can explain antibiotics. Now certain microbes, these are being grown in a particular media which is containing yeast and molasses that is obtained from the sugar industry. Then a particular microbe is being added to this particular media. By growth of these particular microorganism, there is byproduct of the growth of microorganism, it is antibiotic. It is having no use for the bacteria, but antibiotic, as its name suggests, antibiotic, that is, it is acting against the bacteria, other bacteria, that we are getting in treatment part. That is, nowadays antibiotics is the main component of a prescription. If you see any prescription, it contains one antibiotic, and these antibiotics are being obtained from the growth of microorganisms, and that is the nature and source of antibiotic. Antibiotic property is they are not having any chemical interrelation between them. Example is, we can just have the example of antibiotics. First antibiotic which came in therapy was penicillin. Penicillin was isolated by Alexander Fleming while working in the laboratory. He was a microbiologist. So first antibiotic came as penicillin. Penicillin is complex organic acid. It is organic acid. Another example is streptomycin. It is anti-tubercular drug, tubercular antibiotic. It is complex organic base. So there is no correlation between the antibiotics, that is their chemical structure or chemical property. Whereas chloramphenicol is antibiotic which is used in the treatment of typhoid. It is a simple chemical component. Nowadays it is being prepared by chemical synthesis. Initially it was obtained by growth of particular microorganism in this particular media, and we are getting chloramphenicol as anti-typhoid drug. So these are some example of certain antibiotics. They differ in their solubility. Mostly these are insoluble in water, and they require specific dosage form for the use of treatment of a particular disease. Nowadays, the antibiotics which are used in therapy, they also having number of antibiotics are available for the treatment part, and these are nowadays every 2 months there is one new antibiotic in the market. That is, by growing the microorganism, biochemist or microbiologist used to work in the laboratory, and they used to get the antibiotic, and it is being surveyed properly, and then it is put in the therapy, that is the treatment of particular disease. So this is the part. New drugs comes, new antibiotic comes for the treatment part, and these are being used for treatment of different diseases. Now another, by growth of microorganism we are getting enzymes. Enzyme is like, very common enzyme is streptokinase. It is being used for the particular heart disease. Streptokinase and urokinase, these are the two components. These are used to dissolve the clot which has formed inside the body during heart attack. So this is the life-saving enzyme which is given to the patient immediately when there is heart problem, and that streptokinase is being obtained by the growth of microorganism. It is very costly drug, urokinase, streptokinase. Its one ampoule costs about 2000 to 3000 rupees, but it is life-saving enzyme. Now third component, which is nowadays we are getting by modified E. coli growth. E. coli is being grown in the media and we are getting insulin. Insulin is obtained from the pancreas of the animals, mostly of the pigs. Beta cells of pancreas get insulin. But now modified E. coli growth gives insulin. This is in primary stage. If this procedure is successful, then rate of insulin will come down, because modified E. coli growth gives insulin, its rate would be somewhat less. In the treatment of diabetes, insulin is being used, and human insulin is also available nowadays. That is very costly, but it is being considered as good for the treatment of diabetes. So these are the certain example of nature and sources of drugs which gives us the drugs which are being used in the therapy. So this is a complete chapter about nature and sources of drug. I hope it is clear to you.